arrancar. Um, dois, três. Acho que pode baixar um pouco, Fábio. Tá melhor? Mais um pouquinho. 3, 2, 1. Ah, tá bom agora. Testando. 1, 2, 3. Pode baixar um pouco? Pode baixar? Tá bom. Bem-vindos todos vocês a mais uma edição do nosso ciclo de palestras, professor Marcelo André Barcinski. Hoje a gente tem a honra de receber aqui a doutora Christina Sizun, do Centre National de la Recherche Scientifique, que foi convidada pelos alunos organizadores da 11ª semana de graduação em Biofísica como representante especificamente da IUPAB, né, da International Union of Pure and Applied Biophysics. So, let's welcome Professor Christina, who is a chemist by training and obtained her master's and doctor's degrees in physical chemistry from the University of Strasburg, having worked as a postdoctoral researcher at the Max Planck Institute and later at the European Institute of Chemistry and Biology at the University of Bordeaux. Between the years of 2003 and 2020, Dr. Sizun worked as a researcher at the Centre National de la Recherche Scientifique, CNRS, where she currently holds the position of research director, working at the University of Paris-Saclay. She specializes in nuclear magnetic resonance and has a special interest in the structure and dynamics of proteins and protein complexes, ligand-protein interactions, intrinsically disordered proteins, iron-sulfur proteins, and the structure-function relationship of pathogenic proteins. So, without further ado, please, Dr. Christina, thank you very much for coming all this way, and we welcome you to give your talk.
Thank you very much for this very kind introduction, Pedro. And yes, it's really exciting for me to be here. I will give you a brief overview about what we are doing in our group. So I will talk about my favorite protein, which I don't give the name yet. So my talk is about a protein that is disordered, fuzzy, sticky, and I will present some NMR insights into that. So I first will acknowledge the graduate students who worked on the project, which are Christophe, Nicola, Silvar, and I also acknowledge my collaborators from a virology group, which is at the National Institute for Agronomic Research. So Pedro already told you, initially I came as a representative of IUPAB, so the International Union for Pure and Applied Biophysics, to give a talk about, well, what is biophysics today in the world. So I was kindly invited by the Biophysics Week organized by the students, and I must say the Monday was really great. So I'm coming from France. From the University Paris-Saclay, which is quite recent because it fused a number of universities in the area, which is south of Paris. So we have Paris and University Paris-Saclay that I don't show because there's nothing really worth to look at. Our institute, which is the Institute for Chemistry of Natural Substances, which is quite famous because it's where docetaxel was synthesized, which is a drug, a well-known drug, FDA-approved drug, anti-cancer drug, which is sold by Sanofi Aventis. And this drug also contributed to a relative wealth of this institute. And with this money, we were able to afford some very nice NMR spectrometers. So we are now a national high-field NMR facility, and we have a number of spectrometers ranging from 600 megahertz to 950 megahertz. And yes, we are part of this national infrastructure called Infranetics. Now, coming to our topic, I'm working on proteins of respiratory syncytial virus, and I will call it RSV. RSV is an important etiological agent for pneumonia in young children. And in fact, pneumonia is the leading cause of death among children younger than 5 years. So this is quite impressive. Now, RSV was discovered a while ago. It is a ubiquitous virus since all children have been infected by the age of 3 years. RSV is responsible for bronchiolitis. In adults, in general, symptoms are only mild, but it is also very important for immunosuppressed adults and elderly, and it is a risk factor. RSV is a single-stranded negative sense RNA virus that belongs to the Mononegavirales order, Pneumoviridae family, and Autopneumovirus genus. So it's here on the phylogenetic tree. The gene of RSV comprises 10 genes that encodes 11 proteins, and these fall into briefly 4 categories. There are 2 non-structural proteins that participate to unique system of immune response evasion. So these proteins are interferon antagonists. Then there is a matrix. So RSV is an enveloped virus. There's a lipid bilayer, and there is a matrix protein that coats the inner layer of the membrane. Then there are some surface proteins that form some envelope spikes, and these are 3 glycoproteins that are responsible for attachment, fusion, and, well, some other functions that are still unknown. And then there is a whole group of proteins that we are interested in that are responsible for the complex, the RNA-dependent RNA polymerase complex that carries out replication and transcription. And here we have 5 proteins, a large protein called L, that carries out all the catalytic activity. There's a nucleoprotein, and this nucleoprotein is encapsulating the genomic RNA, forming these nucleocapsids that you can see here, and which here are located inside the virion. Then there is an essential cofactor of the L protein called phosphoprotein, and 2 regulators. Either there is the M21 transcription regulator and an M22 replication regulator. Now, of course, we are interested in the way these proteins function, but we are also aware that the virus cycle offers a number of pharmaceutical targets, and the main target is the entry of the virus into the cell with membrane fusion. So there are a number of fusion inhibitors that have been proposed so far, but there is also a second target, which is cytoplasmic, cytosolic, inside the cell, and these targets here are viral replication and transcription. And to inhibit these functions, one has to design viral RNA polymerase inhibitors. Okay, so what is the therapeutic arsenal against RSV today? There are 2 options, prevention and treatment. For prevention, well, up to recently, there was only some prophylactic antibody available with a very low benefit called palivizumab. Now there's another antibody that was approved in 2022, and there are still no vaccines, but in this year, at least 2 vaccines should come out by GSK and Pfizer. Concerning treatment, there's nothing. There's only ribavirin, which is now not used any longer, only in very acute cases because of its toxicity. So if we go back, what are strategies for targeting RSV replication? Now replication, not the fusion process. Well, we can have silencing by RNA to target viral protein mRNAs. Then if we want to target the RNA polymerase, we can either target the catalytic activity, and so this would be done with nucleoside analogs, such as ribavirin. But there is also one option, that is to target protein-protein interactions. And this can be in between viral proteins or between viral proteins and cellular proteins. And well, one can imagine that this could be done using small compounds or peptides. But in any case, there is a need for structural and functional information so that rational drug design can be carried out. And now, if you come to viral RNA synthesis, one has to be aware that this is a dynamic process, since this polymerase complex here, which is mainly composed of the L protein, is elongating new viral RNA using the genomic RNA as a template. As I told you, the genomic RNA is fully covered by the nucleoprotein, and this large polymerase unit needs a cofactor, which is a phosphoprotein. This is why it's called P protein. This P protein is essential because it allows to tether the L protein to the nucleocapsid via interactions between P and N. So here we have an essential interaction, and this is the one we would like to target. Now, for your information, there is a second way of forming an NP complex, and this is called an N0P complex, because the N protein is very avid towards nucleic acids. It needs to be chaperoned, and its chaperone is P. So in the N0P complex, N cannot bind cellular oligonucleotides any longer. Okay. And last thing, which is really interesting, and these are results from my colleagues from INRAE, RSV replication takes place not everywhere in the cell, but in some membraneless organelles that are called inclusion bodies, but you could call them biomolecular condensates whatsoever. And these are really seen when in RSV infected cells. You can also reproduce that by transfection, but what you can see is that in these inclusion bodies you have N and you have P. Now, what do we know? I said it's important to have structural information, and you probably understood by now that I'm more from the structural part and my colleagues from INRAE more from the functional part. There are a number of structural information that was obtained over the years by colleagues. So the first structure that was known was that of an analog of RSV nucleocapsid, and these are these ring-shaped complexes where N proteins form these oligomers. So this is an X-ray structure that was obtained in the laboratory of Felix Ray. Then a little later, a structure of rather low resolution was obtained for real nucleocapsids in the laboratory of Pela. Then we waited, we, together with my colleague Stéphane Duquesnoy from Institut Pasteur, we obtained the structures of an RSV mini NP complex, because what we have here is part of the N protein and a very small part of the P protein. But we will use that. I will come back to that later. And then, well, in 2019, some structures started to come out where the L protein became visible, and this had to wait that cryo-electron microscopy started the resolution revolution. So all the community was really excited about that. So this structure was obtained in the McLellan group. Was really excited because it was the first time that L could be observed. I was excited by that because the phosphoprotein could be observed, so I colored it. And now if you look at that, well, you have here a schematic of the phosphoprotein, and we look at the colors, and we see that, well, although the full-length protein was used to obtain the complex, half of it is missing in the structure. So this means that there's still something to be done to investigate the structure of P. You can also see, well, it seems to form tetramers, and, well, here you see 4 protomers, but the yellow part, you can only see 1 protomer. So, okay. Now, the phosphoprotein is really an interesting protein because it is multifunctional and multi-partner protein. P binds well to a number of RSV proteins, and I already told you it binds to the large catalytic subunit. This is essential. It also binds to the N protein, and also told you in 2 binding modes. Then it can also, it will come back. It also binds to the M21 protein, and M21 is really essential for transcription in RSV. This is a transcription anti-termination factor. And then recently we found out that P also binds, so all the 3 proteins here participate to transcription and replication. So they are part of the large polymerase complex. But P also interacts with a protein that has nothing to do with replication, which is the matrix protein. And in fact, P participates in viral assembly and budding, probably by helping to bridge between the matrix protein that is covering the membrane and the nucleocapsids. And P also binds a number of cellular proteins. Well, there's the HSP70 chaperone, and this interaction is hypothesized to be involved in nucleocapsids remodeling, so there's not much known about that. I will also talk about an interaction with the cellular phosphatase, which is PP1. There's an interaction that was found with tropomyosin that, well, could be involved in replication, but maybe also in virus assembly and budding. And so the question is, how does P accommodate multiple binding partners? Because P is not a very big protein, but it has multiple partners and probably multiple binding sites. So what is really the structure for RSVP? When we look at P phosphoproteins of the mononegavirales order, they're all organized in the same way. They are oligomers, they can be trimers, dimers of dimers, tetramers, so there's always an oligomerization domain. There is a C-terminal domain that is in general involved in nucleocapsid binding, and then there's an N-terminal domain here, and this one is thought, no, it's not thought, it was shown in most cases to be involved in forming this N0P complex. Now, what is known about structures? Maybe there are probably some else now because this slide is already old. So there are structures of several domains of phosphoproteins, and you see there is some diversity inside the structures. And this is important because you see here we have the filoviridae, we have a trimer, tetramer, quite long. Here, well, if you look here at VSV, the dimerization domain looks very different. So it means that you cannot infer structure knowing one virus for another. And the same goes here for the nucleocapsid binding domains. They're all different. So it means that there are really different binding modes, even if the domain organization of this is similar. The detail then can be quite different. So the questions we asked was, well, what are really the domains inside RSV phosphoprotein? There seems to be disorder, and disorder was predicted, so what is the degree of disorder? And then, well, where are the binding sites, and what is their size? So, since we have a nice NMR facility, we are using NMR quite a lot. And I show you here this typical 2D spectrum, which is called an HSQC spectrum. I know that a number of people here are familiar with NMR, so, but it's for those who are maybe not. So this is called this HSQC spectrum. This is very important because it's sort of fingerprint of a protein. And what we can see here is that there are a number of peaks. They are sharp, and they are not very well dispersed. And this in general indicates disorder. So this protein, NMR, straightly indicates that RSVP harbors a number of intrinsically disordered regions. And then there's something else which is nice about these spectra is that the number of signals corresponds approximately to the number of residues. So if you count the number of peaks, then you should have the same as the length of the protein, and here it's not the case. 1/3 of the signals is missing. These are broad, so this points to regions that obviously are not disordered, or maybe not disordered, but rather ordered. And one of these ordered regions was identified, and it's a small region here, about 40 amino acids, and this is the tetramerization domain. So the structure was solved by, I would say, a number of techniques. We did this by NMR, mostly for fun, because it's not very easy to obtain the structure of a tetramer, but Nelson managed to do so using some specific isotope labeling by using SACs, so small angle X-ray diffraction data. We were able to show that, well, this small construct here was compatible with the tetramer. So this seems to be, this is one of the structured regions, but it does not account for 1/3 of the protein. So the beauty of NMR is that you can go further, and you can in fact measure chemical shifts for each of the residues, at least those you can see, and you can also measure carbon-13 chemical shifts, and from these shifts you can predict secondary structure propensity. And if we look at what we observe for phosphoprotein, well, you see in white this is disordered, in red we have the percentage of propensity for alpha helix. So it means that, well, what we can see is really disordered. Only this region here seems to have some alpha helical propensity. It's quite small. Now, this is supposed to be the N0 binding region, and at this point, the structure of N0P for another virus was solved in the group of Grimes. And here, so what you see in gray, it's the N protein, and what you can see in orange, it's, well, just this part of the phosphoprotein of HMPV, and it forms a helix when it forms the complex. So it's not the full N0P complex, but here we have really the binding epitope. Now, there's something, so this means that this alpha helical propensity here seems to indicate that when the complex is formed, an N0P complex is formed for RSV and P, then this helix is stabilized. Recently, we looked at the phosphoprotein of HMPV. So HMPV is human metapneumovirus, so it's of the same family as RSV, same symptoms, pneumonia, infection of the respiratory tract. This is really interesting because we have here helical propensity for a protein that is really cognate. And what we can see is that in the free form of P, now this helix for HMBV free of P is fully formed. So this may also give maybe an indication about, well, we cannot talk about affinity, but the way this complex is formed, or maybe it can also explain why my colleagues from INRAE really tried hard to get this N0P complex for RSV, and they never managed to crystallize it. Seems to be possible for HMPV, but not for RSV. Maybe because there is some some flexibility still that makes the complex more difficult to form. Okay, so we have more rather disordered regions here, especially this whole N-terminal region, which I call PNT, and here this tail region. We looked at that by 15N nuclear relaxation. So these are dynamic parameters that can be measured using 15N labeling. And it's interesting because this shows you that, well, this region here that has this very low alpha helical propensity has some specific behavior, and this here, this increased R2 means that there is some conformational exchange taking place. But then there's also something happening. So here we have this region that we cannot observe, and something is also happening here. And this is interesting because, well, it's difficult to say that for just using the chemical shifts that something is happening here. So at some point we will come back to these regions that are proximal to the oligomerization domain, which is right here. We were quite unhappy with the fact that there's still a region missing here. This one, well, I term it PC alpha, and you will know in a few minutes why it's called this way. We badly wanted to look at this region, so it's impossible to look at it using full-length P, and so we started using fragments. And one of the fragments that we used is one where the oligomerization domain was deleted. So could give us some indication. So if you count the peaks on these shorter versions, there are more peaks than on the full-length spectrum. And if we look again to relaxation data, and not only to relaxation data, now this part here now can be observed. So this is good news. And we can also see that, well, there's really increased R2 relaxation taking place in this region. And we already said here, well, this is a region with alpha helical propensity. So let's see if this correlates with secondary structure propensity. So here is the construct P delta OD that we used for relaxation. And now you can see that in this region, this is an alpha helical region, maybe with two different alpha helixes that are nearly fully formed. So this is quite surprising, because one would expect to see these helices in the spectrum of full-length P. If they are completely broadened out here in the full-length protein, this means that there are probably some conformational exchange, but they can also be transient contacts, which would contribute to this phenomenon. And well, if you look at other fragments, these shorter fragments are all compatible with what we observe on P delta OD. Okay. Now, if we compare these results, you could argue, well, here you use a protein that is monomeric and not tetrameric. Does this represent the full-length protein? This is a real question. When we compare that now to the cryo-electron microscopy structure obtained for the LP complex, this region here, now called PC alpha, because it's C-terminal and has high alpha helical propensity, it has also high alpha helical propensity within the structure. So this show you something. What is interesting is that each protomer here adopts a different conformation. So this also means that the helices cannot be fully formed or should not be fully stable because they have to adapt, their conformation has to adapt when it binds to L. So to get a sort of picture of RSV phosphoprotein, it might look like this. So the small tetramerization domain here, well, these transient alpha helixes at the C-terminus and all the rest, which does not adopt any structure. So this is a little bit biased here picture because we used a software called Cyana, which makes more extended structures. You would probably expect, well, this part to be a little bit more coiled up. Now, we were wondering, yes, about compactness of RSV P protein. And here we used a very nice tool used paramagnetic relaxation enhancement, which results in line broadening, which means that you lose signals in the vicinity of a paramagnetic probe. So this will report on transient long-range contacts up to 15 angstrom. To do so, you have to have a protein with a cysteine. So in the case of RSV phosphoprotein, there is no cysteine in the sequence, so it has to be mutated. But so we mutated a number of serines. Sometimes it was a glutamate, and we added this nitroxide label. So if you add a nitroxide label at position 23 here, you have some broadening. So here we measure the remaining intensity. And this is normal because it's in the vicinity of the paramagnetic tag. But you can also see that you have some broadening, long-range broadening. And this would indicate that there are transient contacts between this, well, alpha N1 region and this region. And if you remember, this was the region that was transiently structured, very transiently structured as an alpha helix. And this is the region where we had increased R2 relaxation. And what is also important to notice is that here, if this region here adopt a fully alpha helical conformation, then they will expose a hydrophobic phase. And it's a little bit less pronounced here, but this could mean that there are some transient contacts that are mediated by hydrophobic contacts. So we did these experiments for a number of positions of the paramagnetic label. And so if we represent them on the structure here, we can see that there are a number of contacts that take place between different parts of the protein. And in general, these parts here are quite sticky. The fact that when you have a label on the N-terminus, you never affect the C-terminus seems to indicate that the tetramer of P is parallel. It's something that we did not know when we did these experiments. So we get some extra information. Then we were interested again, yes, in this PC alpha region. For NMR, it's always, well, we start with these 2D spectra, and we had to use fragments, as I told you, to look at this region. So we were looking for the shortest possible fragment. So we did a number of constructs that contain this time the oligomerization domain. So we really wanted to have a construct that is more meaningful. And you see that, well, when we take a very long construct here, we completely use this region. If we use, yes, even this construct here, we are not able to observe POD. Here, yes, even here, you don't. We don't manage to see the signals all the signals using this constructs and this temperature. In fact, this temperature is used for intrinsically disordered proteins, so we increase the temperature. In general, this is more favorable for globular proteins, and in this case, we were able to recover the signals. And I will show you something about this construct here. So we were able, now working at 330K, to observe all the signals of our fragments. And here we have this fragment that, well, we have a short fragment, a long fragment. And the long fragment here really reproduces what we would expect, the tetramerization domain with fully formed alpha helices, and here some, well, nearly fully formed alpha helices. But when we take a shorter construct, where we thought we had deleted the second helix, then you can see that, well, the first helix here is not fully formed. So the way you are designing your constructs is really important for NMR to get meaningful results. Here again, we applied PREs now to see if there are contacts between this alpha helical C-terminal domain and the oligomerization domain. We used two positions, and you can see here we put the paramagnetic tag onto the tetramerization domain, and we are able here to see some transient contacts with the alpha helical domain, and well, the same if we use this position. So this domain really transiently binds to POD, and this might explain why it is so difficult to observe it in the full-length protein. Now, this is for Fabio's group, because this is about interfering with the flexibility of a protein. It was one of the first experiments we did. We had used here a construct that did not contain the oligomerization domain, but which is the C-terminal domain of P. And the group of Jean-François Leouet had found out that garcinol could maybe inhibit the RSV and P interaction. So the idea was rather that garcinol, this molecule here, binds to N. But then, since we had some P left and there was some garcinol, we just looked at the mixture. And what we saw is that by adding the small molecules, we were indeed able to reduce the intensity of all this C-alpha domain here, which means, in principle, when you see that, you would say, well, first there is some binding taking place, but you would expect binding to be much more localized. Here you have a global effect. And we did the same here using a tetrameric construct. And again, by adding garcinol, you just broaden out a full region, a very large region, and only the flexible region here stays as it is. And if we look, well, at the size, because one explanation could be that garcinol triggers some oligomerization or some aggregation. It does a little bit, but it does not really fully explain what happens here. So it seems that there are some molecules that could interfere with the flexibility, the remaining flexibility of the C-alpha domain. And as I showed you, or if you remember the cryo-EM structure of the LP complex, this region is important for binding to the L protein. So maybe by interfering with dynamics of P, you are able, and this is something, just an idea, it has not been proven, you could maybe interfere with formation of the PL complex, because flexibility is necessary to form this complex. Now, P is disordered. It has a number of partners. The question is, well, is there some folding? So I will show you two examples. One example is the P-M21 interaction. As I told you, M21 is a transcription anti-terminator that is recruited by P to this inclusion bodies. M21 is really interesting because it binds to P and to RNA in a competitive manner, and Fabio knows a range about that. So we are interested here about the interaction between P and M21. So what we do is we use labeled 59 labeled P. So here we have just a zoom on the 2D spectrum of P, which is in black here in the free form. And when we add the domain of M21 that binds to P, then the signals disappear. And they disappear here in this region, which is the region that had this increased R2 values. So here we have the binding region of M21 on P. So it's a linear binding site. It's difficult to say if it's really small, but it's not large. We did the reverse by looking directly at M21, and we were able to delineate here binding surface using some experiments called transferred cross-saturation. And using all of this and some mutagenesis data, then we were able to model the M21P complex. And in fact, yes, P needs to form a helix here. So this region that had a very, very small alpha helical propensity would be stabilized inside the complex. And this was further later confirmed by X-ray diffraction in another group. Okay, so RSVM21 binds to a small region of P with, a small region of P with low alpha helical propensity, but in the end forms this fully formed helix. And then a second example, cellular phosphatase PP1 alpha binds to P. So this has been shown by pull-down experiments and other binding experiments. Now, when we look, we do some NMR to have some atomic resolution information. What we can see is, this PP1 alpha, so now you know that we have to look at the intensities. It does not bind to this region, it seems to bind to the N-terminus. And, there are two regions that can be seen that have low intensity when we add PP1 alpha. And here in this region, we find a motif that is called to be a PP1 binding motif, which contains this lysine, this valine, this phenylalanine. So it's not completely the canonical sequence, but it's there. So the complex formed between P and PP1 alpha is meaningful. And just by modeling using some homology modeling using structures that are known for PP1 alpha, then we could see that we have this small stretch here. So again, a small linear binding site that docks here onto this existing beta sheet. I told you about binding of two proteins, and in fact they are linked, and this is important. Because M21 is a phosphoprotein that is phosphorated and dephosphorated in a cyclic manner, and this is important because when it is phosphorylated, it cannot bind to RNA. So once it is unphosphorated, then it can bind to messenger RNA. And it was also shown that dephosphorylation is being made by the PP1 phosphatase. The fact that PP1 and M21 do not form any complex on their own. So in this case, the P protein is needed as a platform to accommodate both partners and to bring them into close proximity. And so M21 binds probably to one protomer on one side, PP1 binds to another protomer, to a very proximal site, all on the N-terminal part of the protein. And in this configuration, M21 can be dephosphorylated. Now, I have 2-3 minutes to talk about the P N interaction. So we already said this is important. So the phosphoprotein is really the linchpin of this polymerase complex. I also told you there are some structures for N. And well, if you take this RNA rings, we look at the protomer here, you can see that the N protein falls into 2 domains and RNA binds into this cleft. When we were looking for NMR conditions, we said, okay, I cannot work with that. This is too big for NMR. I'd like to have a monomeric protein. It's possible by using mutants, but in general, they're not very stable. And then I said, okay, if we can just have this domain, it should not bind to RNA, and it does not. So we were really looking to investigate the NP complex. We were looking at a sort of minimal complex, and I also already told you, and we found that this minimal complex would be formed of the N-terminal domain of N and the C-terminal domain of P. And my colleagues here from INRAE also made a number of mutations showing that the P binding domain on N here is in this cleft. Now, coming back to NMR, we used these domains that interact with each another, and we looked at interactions. We were quite, so if you make the experiment looking at intensities, then we see that N binds here to N binds to the C-terminus of P. So this was not completely surprising. We were expecting that. Here we have some residues that belong to the C-terminus of T. Since we have, so this is a sort of titration. Oh, there's something missing. Here. This titration here allows us to extract binding constant, and it's 50 micromolar. And you will say, oh, this is quite low affinity. But don't forget that this complex takes place in a process that is processive. The complexes must be formed, but they must also unform so that the polymerase can go on. So it's not completely, so this is not a bias. It's something that we found by other techniques, so 50 micromolar. And yes, just last comment on that one. There's a line binding here. There seems to be spurious binding because you can see that something is happening here. And this is, yes, the case, often the case. So it's more difficult to explain what happens, but, well, it happens. So we tried to construct a model for this PN complex. We used NMR. So by NMR, we used chemical shift variations. So here we looked at the N protein. We added some P peptide that corresponds to the C-terminus of P. We can see some very nice perturbations here. And when we map the perturbations, well, we find this cleft. We're quite happy that this corresponds to mutagenesis experiments. And on the other hand, my colleague Stéphane Duquesnoy at Institut Pasteur did some X-ray crystallography. He co-crystallized this N-terminal domain of N with several P peptides, and he obtained some structures of complexes. And he showed that the last amino acid of P binds into a hydrophobic pocket inside N. Only in the case of this dipeptide here, P2, he was able to see some signal for P. When he took longer peptides, then he only observed this phenylalanine. So it seems that there is really this phenylalanine anchors P onto N, and then the rest is more or less disordered. So, well, we will do this shortly. So, well, X-ray diffraction is really nice because you get this detailed structural determinants. So, hydrophobic interactions are important, but electrostatics are also very important in this case, and this is something that you can see by changing the ionic strength of the buffer used. So X-ray crystallography shows that, well, the main structural determinant is this phenylalanine. And so Felix Ray said we only need this phenylalanine. Now by NMR we did some titrations. I didn't use phenylalanine, but this very short peptide, and well, we measured the affinity. In fact, if you only use phenylalanine or the aspartate phenylalanine peptide, you have millimolar affinity, certainly not 50 micromolar affinity. So in fact, even if this anchor is, of course, if you remove this anchor, nothing happens. It doesn't bind any longer. But if you want to have affinity, this is not enough, and you need up to a stretch of 10 amino acids. And what you can see is that there are many, many negatively charged amino acids. So in fact, it's a little bit difficult to see on this picture here. There is sort of avidity to bind to this part here. So this means that there are, in fact, multiple and specific electrostatic interactions outside of the binding pocket, away from the initial cysteine, from the, well, so 2 amino acids that are quite close. You replace an oxygen by sulfur. At some point it was not possible. When we made the mutants and the tetramerization domain, we had to take care that we chose a surface exposed residue. But by that time, Stefan had already an X-ray structure of the tetramerization domain, and so we were quite sure that we were not disrupting the tetramer. That is important.
So do you believe in secondary structure formation inside the cell, and then it's became the constant binding lower than you see?
This is a good question. Because yes, you're right, we are working on solution, there's only P, maybe it's its partner. It's possible since, I told you, yes, lysozyme seems to bind. So it means that there is some spurious binding, and this, so 2 answers. First, the spurious binding could maybe maintain could shift the equilibrium of the conformational equilibrium more towards alpha helices, if it's alpha helices. Then we thought the fact that there are these internal contacts seem to protect more or less also against some unspecific contacts within the cell. So this is also a hypothesis, but it would be nice, yes, to make some in-cell NMR with this, yes, and to see what changes. This would really be something nice to do.
Thank you for the talk, Christina. And I'm just wondering if the inclusion bodies environment that you have mentioned have any relationship with liquid-liquid phase separation, and if that contributes somehow to the high values of the dissociation constant with the partners.
So you're right. So it does, yes. Liquid-liquid phase separation is probably triggering formation of these inclusion bodies, since it's possible to get this biomolecular condensates just by using N and P and some crowding agent in vitro. Of course, then it should be shown that it is the same, but it seems to be really the physical explanation for that. This means that N and P are always in close proximity, and this would also shift, of course, equilibrium. It would not shift equilibrium, but the avidity then would be different. So K on and K off values would be affected, and so probably also KD.
Christina, I was going to ask the similar question, but I'm curious about the content of N and P in these inclusion bodies, in these liquid condensates?
So it seems the majority, since, well, N is the most abundant protein, P is also very abundant because after the non-structural proteins, these are the first replication complex proteins that are synthesized, so they're really abundant. Then in this foci, you will have L protein that is found. You will find M21. I cannot tell you what the percentage is because nobody never looked at that, but from the amount of proteins that are in the cell, it's mainly N and P and some L, some M21, and some of all the cellular factors. It's also where you will find PP1. You will have co-localization between P and the matrix protein. So everybody, yes, participating into the viral cycle is in these inclusion bodies. Yeah.
Okay, so I actually have many questions myself, but I selected 4, and I will ask them in 2 batches. So there are 2 more specific questions, which are about your work, like more directly, and 2 more general questions. The first question would be regarding the formation of the alpha helices on the C-alpha domain when you removed the tetramerization domain. Why does it happen? Like you remove a region and then you increase the propensity to form helices in the next region?
No. In fact, when I remove the tetramerization domain, I can see them. But of course, they're not bound to the tetramerization domain. And under some conditions, yes, I can see them. So at high temperature, in fact, I can observe them. And also the tetramerization domain. And so they are formed with and without the tetramerization domain.
You just cannot detect them.
Yes, it's a problem of detection. That's always the problem of NMR, is as soon as you start having transient contacts, then you lose the signals.
Okay. Question number 2 is regarding the lysozyme. Did you manage to get where it binds in the P protein?
Yes, it binds on the M21 binding site.
And now for the 2 more generous questions, I'm just going to ask them together. What do you think makes a protein intrinsically disordered, and what do you think makes proteins sticky?
So, what makes intrinsically protein disordered? It's really a matter of sequence and amino acid composition and sequence of following. This has been demonstrated that for intrinsically disordered proteins and regions, there is a bias in amino acid composition. So this is completely physical.
Are they charged?
Yes, more charged residues, so less hydrophobic residues. Absolutely, can be also many polar residues, but there's a real bias. And then what makes sticky? At some point, you can have some more hydrophobic patches. And since it's not, there are no secondary structures that are fully formed, these patches are exposed. And so they could just stick to each other.
Okay, thank you very much. If there are no more questions, then we'd like to thank you once again for coming. And, well,
thank you, Peter.
Feel welcomed.
Thank you.
Thank you, everyone.
Thank you for listening, for discussing.